Muscle contraction cycle. During the contraction of a muscle, the sarcoplasmic reticulum, you could think of it as a specialized form of the endoplasmic reticulum or as a network of membranous tubules. Or to simplify its appearance, you could also think of it as a little net that wraps around the myofibrils, also known as muscle fibers. So the sarcoplasmic reticulum releases calcium ions into the sarcoplasm. The sarcoplasm is inside the muscle fascicles. Here they bind to troponin, which is a protein. Troponin then moves tropomyosin, a protein responsible for muscle contraction. It looks similar to a rope around the actin. The tropomyosin moves away from the myosin binding sites on the actin. Once the binding sites are free, the contraction cycle starts. Step 1. ATP hydrolysis, or creating energy and turning on the myosin head. The myosin head, or the moving fist, involves an ATP binding site and an ATPase, which is an enzyme that hydrolyzes or breaks down the ATP into ADP. Or ADP is called adenosine triphosphate, and there's a phosphate group as well. And that is a molecule that is the byproduct of ATP that has been hydrolyzed to provide energy to the muscle contraction. Now the myosin head is prepared, it is loaded, and it is in a high energy state. Step 2. Attach the myosin to actin to form cross bridges. The energized reorientation of the myosin head, or the fist, is ready to attach to the myosin binding site on the actin, and it releases the hydrolyzed phosphate group that we talked about in step 1. The release of this triggers step 3. When the myosin heads attach the actin during contraction, they are called cross bridges. Step 3 is the power stroke. After the cross bridges form, the power stroke starts. The site on the cross bridge where ADP is bound opens as a result. The cross bridge rotates and releases the ADP. The cross bridge generates force and it rotates towards the center of the sarcomere, adding the thin filament past the thick filament towards the end line. The M line is in the middle of the filament. Step 4. Detachment of myosin from actin. When the power stroke is finished, the cross bridge remains attached to the actin until it binds to a new molecule of ATP. As it binds to the ATP binding site on the myosin head or the fist detaches from the actin.